Okay, so welcome back to this next video in which we are discussing the endocannabinoids in the cardiovascular system. Okay, so we've seen how there is no endocannabinergic tone, okay, i.e. the endocannabinoids do not seem to be involved in normal homeostasis of the cardiovascular system, okay. Instead, their role seems to principally be in pathophysiology, and we think they have a very important role when tissues become ischemic, particularly if the heart becomes ischemic. Okay, we think that tissues that are ischemic may produce endocannabinoids, and when you get a local production of endocannabinoids, what that can then cause is vasodilatation of the local blood vessels leading to that tissue, and therefore increased blood flow to that tissue, and therefore help to reverse the ischemia. In particular, if this occurs in the heart, then not only will it increase blood flow through that mechanism that I've just stated, but also it actually has a negative inotropic effect on the cardiomyocytes, okay? The cardiomyocytes have CB1 receptors on their surface, and this produces a decrease in the contractility of the cardiomyocytes. So basically you make the cardiomyocytes work less hard and use less oxygen. Again, that's beneficial if if this portion of myocardium is suffering from ischemia. Okay, so that's where this is going towards, okay? We've then seen what happens if the endocannabinoids go up systemically in a conscious animal. Okay, we've seen that then what happens is something very different. What happens is in the brain you increase the central sympathetic output, okay, and this increases the sympathetic output to the heart and to the blood vessels, and what this does is overpowers the other effects of endocannabinoids that are more local and causes the complete opposite. Okay, it causes the heart rate to go up, it causes the contractility of the heart to go up, and it causes blood vessels to constrict. Okay, so the rise in sympathetic tone from the central nervous system then opposes the local effects of the endocannabinoids, which are aiming to do the exact opposite. Okay, right, so what I'm now going to discuss is one final experiment which is interesting, okay, because there is a way of blocking the rise in sympathetic tone that is generated centrally in response to endocannabinoids, and this is to use a general anaesthetic. Okay, so instead, if you do this injection of anandamide into an anaesthetized animal, okay, I've just got to concentrate on spelling anaesthetized properly. I think that's the correct spelling of anaesthetized. Okay, so if you do this experiment in an anaesthetized animal, which is one that has been injected, let's say, with a general anesthetic, and then basically the result that you get is very different, because the general anesthetic blocks the rise in sympathetic tone that is generated centrally due to the systemic injection of the endocannabinoids. So now let's have a look at the graph that you get for mean arterial pressure if you inject a bolus of anandamide into the circulation. Okay, so this is time on the x-axis and this is mean arterial pressure on the y-axis. So basically you get a graph that looks like this. This is mean arterial pressure initially at 90 millimeters of mercury, and then you get the same effect as before. You get an initial depressor effect, which again is the Basalt-Jarisch reflex. Okay, and that's known as phase 1 of this graph. Okay, and we're going to see that there are 3 separate phases to what happens when you inject anandamide into an anaesthetized animal. Okay, then what happens is you get a phase that no one can yet explain as far as I'm aware, which is phase 2, where it temporarily goes up, and then you get phase 3, which it shouldn't quite go down that low, but it gets the point across at least. Okay, which is this depressor effect, and this prolonged depressor effect where blood pressure goes down. So this is called phase 3 and this is called phase 2. Okay, so phase 2, as I said as we were going through it, we can't explain this. Okay, you get this temporary short-lived pressure effect and no one knows exactly what actually causes that. Okay, But then this is the interesting portion. You do get a prolonged depressor phase which lasts for tens of minutes. Okay, so this is the portion that we might predict now from our understanding of how endocannabinoids work within the cardiovascular system. Okay, so just to go over this then, so when you initially inject the anandamide in, again it triggers this Bezold-Jarisch reflex. It activates the C fibers in the walls of the myocardium by activating the TRPV1 receptors on those, and that then goes up to the brain and increases the parasympathetic tone to the heart, which causes a bradycardia, and that causes blood pressure to go down. Okay, we'll then ignore the phase that no one can explain, and we'll go on to this phase 3. Okay, in phase 3, what happens? Well, now you've got the systemic endocannabinoids, so what happens? Well, those are going to cause vasodilatation at loads and loads of blood vessels, okay, by the mechanisms that we discussed here. Okay, so it's going to decrease sympathetic tone now because the cannabinoid 1 receptors are going to decrease the amount of noradrenaline being released by these sympathetic axon terminals. Okay, you're also going to get the release of the calcitonin gene-related peptide, which causes the hyperpolarization of the vascular smooth muscle cells. Okay, and also the direct effect on the CB1 receptors on the vascular smooth muscle cells and the endothelial cells, and both of these again lead to hyperpolarization of the vascular smooth muscle cells. In the case of the endothelial cells, it's by the release of the endothelium-derived hyperpolarizing factor. Okay, and this time we don't need to worry about sympathetic tone going up because of the central effect, because the general anesthetic has blocked that. So the overall effect now is vasodilatation, and therefore in this phase total peripheral resistance is going to be going down. Okay. In addition, if we think about what the effect on the heart is, firstly, you decrease sympathetic tone to the heart because of the CB1 receptors on the sympathetic nerves that innervate the heart, okay, which will decrease sympathetic tone to the heart by decreasing the amount of noradrenaline that those fibers are going to release in response to each action potential. That decrease in sympathetic tone is going to lead to bradycardia, decrease in heart rate, and also a decrease in contractility, which again is going to lead to a reduction in cardiac output. So in this phase, cardiac output is also going to drop. In addition, remember the direct effect of the CB1 receptors on the cardiomyocytes, which is a negative inotropy effect, so that also decreases contractility. Okay, so without the rise in central sympathetic tone due to the cannabinoids, because we blocked that by the general anesthetic, basically a systemic injection of cannabinoids does produce a prolonged depressor effect, basically, which is caused by total peripheral resistance going down and cardiac output going down, which are the two things which control mean arterial pressure. Okay, right. The other thing to say that is notable is that there is a drug, okay, and this drug is rimonabant. Okay, now rimonabant is an anti-obesity drug, okay, and the reason it's an anti-obesity drug is that it has some effect on appetite. It has an ability to decrease appetite, okay, for reasons that aren't particularly well understood. It's not particularly good at doing it, but it is used as an anti-obesity drug. Now, rimonabant is a type 1 cannabinoid receptor antagonist. Okay, and it's believed to be the antagonism of CB1 receptors in the brain that is believed to underlie its decreasing appetite effects. Okay, right. Now, the other thing that I should just say about rimonabant is that before it was actually approved for use in obesity, okay, it was an experimental drug for a lot longer period than it was a clinically used drug. Okay, so if you read the literature, you will often hear it referred to by its original code name. So before drugs are actually used clinically, they aren't given a fancy name like rimonabant. Instead, they're given a code name. Okay, and the code name for rimonabant was SR1411716. Okay, so if you read literature on the endocannabinoids, you will often hear rimonabant referred to as SR141716 rather than rimonabant. As I say, this is because it was used as an experimental drug long before it was ever used clinically. Okay, and it was known that it was a CB1 antagonist. Okay, right, now basically this drug, rimonabant, if you give this to this anaesthetized rat, which we're going to inject the anandamide into, it gets rid of phase 3 basically. This is abolished. Okay, so you no longer get phase 3 if you inject rimonabant as well. Okay, now what does that show you? Well, that probably shows you that phase 3, this depressor effect, is principally caused by CB1 receptors. Okay, now there is a little controversy there because rimonabant actually binds to loads of other things as well. So it's a bit of a claim that it's due solely to its antagonism of CB1 receptors, but it's unlikely due to that. Okay, so what does that suggest? Well, let's go back to these effects. Well, that's absolutely fine because most of the effects of anandamide are caused by CB1 receptors. So look at it, most of this is all about CB1 receptors, but this TRPV1 mechanism, that's not going to be blocked by rimonabant. Okay, so what that suggests is that this TRPV1 mechanism isn't that important, okay, or potentially this is only important in very few blood vessels is the conclusion there. Okay, so the TRPV1 mechanism may be important in just a few blood vessels, for instance the rat mesenteric artery, which is one of the ones that's principally used in this investigation. Okay, because if it wasn't the case that TRPV1 wasn't very important, then you'd expect there still to be a bit of a depressor effect even when the rimonabant was given, because the rimonabant isn't going to affect the effect of anandamide on the TRPV1 channels. Okay, right, so that's a nice little confirmation of that. Okay, right. So now what I want to do is I want to summarize where we have got to with this, and then I want to take it further by looking at the CB2 receptors and their role in potentially protecting you against ischemia reperfusion injury. Okay, so to summarize then, what we have seen is that if you get a local rise in endocannabinoids, okay, so a local rise in endocannabinoids, what this causes is it causes vasodilatation to that local area and therefore can protect you against ischemia. Okay, protects against ischemia. Okay, and this is probably particularly important in the heart because the endocannabinoids not only vasodilate the blood vessels supplying the myocardium, but also in ischemia in the heart, what they can also do is decrease the force of contraction of the cardiomyocytes and therefore decrease the oxygen and glucose expenditure of the cardiomyocytes, and that's also pro-protection against ischemia. So, the role of the endocannabinoids then is probably that they're released in this pathological situation of ischemia, and there are loads of other inquiries about how they're released in other cardiovascular pathologies, not just ischemia, but it's ischemia that makes the most sense as of yet, so that's the one that I'm presenting principally. Okay, right, now I'm going to tell you about the role of the CB2 receptors in ischemia reperfusion injury, and this potentially starts to fit the two stories together, okay, and make this make sense. Okay, right, so CB2 receptors, we haven't seen them at all yet in the cardiovascular system. So type 2 cannabinoid receptors, where are they present? Well, they're present actually in a lot of places. Okay, they're present on the endothelial cells, okay, which I'll abbreviate down to ECs. They're also present on the myocardial cells themselves, so they're present on heart cells. Okay, they're also present on other peripheral tissues as well. So for instance, liver cells have CB2 receptors on them, brain cells have CB2 receptors on them. Okay, and in addition, one of the really important places where CB2 receptors are found, which was initially considered the only place that CB2 receptors were found, is on immune cells. Okay, and this is going to be very, very important. So they're found on cells of the immune system, and this initially was considered the only place that CB2 receptors were found. Okay, right, and their role seems to be anti-inflammatory, preventing inflammation. Okay, so let me just outline briefly the inflammatory response, because we're going to need it to understand the ischemia reperfusion injury and how this can be protected against using the CB2 receptors. Okay, so basically, in all the tissues of your body, you have special cells which are known as sentinel cells. Okay, and these sentinel cells, their job is to stand guard. So let me draw one of these. Okay, so they're just distributed among the normal cells of that tissue. Okay, and their job is to stand guard and look out for pathogens. So let me give you examples of sentinel cells. So one of the key examples of a sentinel cell is a dendritic cell. Okay, and this is what I've drawn here. Okay, now dendritic cells have absolutely nothing to do with neurons. Okay, they just have dendrites, but they are not capable of conducting action potentials. Okay, so they have this star shape, but they're nothing to do with neurons, completely different cells. And I'll give it a nucleus because it looks a little bit odd without one. Okay, so they're one of these sentinel cells that is standing guard and waiting to respond if a pathogen should arise. Okay, other examples of sentinel cells are macrophages. Okay, so you have some macrophages known as tissue macrophages, which are normally present in tissues and are sentinel cells. They're standing guard waiting for evidence of an invading pathogen. Another example of sentinel cells are mast cells. Okay, now these principally release the molecule histamine, but they can release other pro-inflammatory mediators as well, and again they're distributed all over the body. Okay, right. Now, what do these sentinel cells do? Well, basically, their role is to respond if a pathogen should arise. So let's say there is some horrible pathogenic cell amongst all of the human cells. So most of the cells in this tissue are human cells, but then suddenly there's this little bacterial cell here. Okay, and this bacterial cell is a threat. Okay, a big threat. It could start dividing and dividing and dividing and dividing, and these bacterial cells could start chucking out all sorts of toxins which could kill the human cells. So this is a big problem. Okay, basically the sentinel cells have all sorts of mechanisms for detecting the presence of these bad invading cells, okay? And when they get evidence that some horrible invasive pathogen is present within the tissue, what they effectively do is they call for help, okay? They scream out for help, basically, from the immune system, okay? And the fancy word for their screams is pro-inflammatory cytokines. So they release all sorts of molecules that are basically just calls for help, okay, which we're just going to summarize as pro-inflammatory cytokines. Now, where is help going to come from? Well, it's going to come from the cells of the immune system, which are specialized cells which are capable of destroying pathogens. But where are all of these cells? They are not normally distributed amongst the tissue. Instead, they are within the blood. Okay, so let's have a blood vessel here, like so. So this could be a capillary maybe, or potentially a post-capillary venule. So here are endothelial cells lining this blood vessel here. Okay, so what we want is help from the immune system. Okay, so what's going to actually happen is you need to bring things out of the blood into the extracellular fluid of this tissue in order for them to fight the pathogen. And I should just say something that I missed a moment ago. The immune system doesn't just consist of the cells of the immune system. There are other components of it as well. For instance, the complement cascade is a very important part of it. In addition, the fibrinogen cascade is an extremely important part as well. Okay, and these are proteins that are in the blood which can help kill the pathogen. But the important thing is that the components of the immune system are within the blood. Okay, so basically, if we're now going to get these components out of the blood into this peripheral tissue which needs their help, we need to open up the gates to them, and that's the role of the endothelial cells. So basically, the pro-inflammatory cytokines work on the endothelial cells here, and they cause changes. Now, what are the principal changes they cause? Well, they cause what's known as endothelial contraction. Now, let me show you this in a picture. Okay, what's going to happen is the endothelial cells initially, you know, they're nice and tight. They're covering up the surface of the blood vessel, and they're sealing this. This is a nice tight blood vessel. Okay, there's not much leakage. Whereas now what's going to happen is the endothelial cells are effectively going to contract down like so and make large holes between them, okay, out of which fluid can leak, and more importantly, proteins can leak. Okay, so this is what's known as endothelial cell contraction, or just endothelial contraction for short. Okay, right, and what this allows is it allows fluid from the blood plasma along with proteins that are in the blood to move across the endothelium into the extracellular fluid of this tissue. Okay, so now what happens is fluid accumulates in this tissue, and you get swelling of this tissue, and this new fluid that's that's accumulating in this tissue is known as an exudate, or more strictly, an inflammatory exudate. Okay, now why is this helpful? Well, within this inflammatory exudate you have all sorts of proteins that are capable of attacking the pathogen. Okay, so some of these are complement proteins, for instance. Okay, so complement proteins, which are actually capable of attacking the pathogen themselves. Okay, they respond to certain molecules that pathogens have and then start doing horrible things on the surface of those pathogens. Okay. In addition, there are proteins that can start off the coagulation cascade here, or what I previously called the fibrinogen cascade. And basically what happens here is you start making a mesh of fibrin, okay, a mesh of fibrin all over the place. So you start producing these fibrin strands from proteins that are within the blood, and this makes a fibrin mesh. And the idea here is that you can trap the pathogen in and stop it spreading into the blood, because the last thing you want is it leaving this area of tissue and spreading elsewhere. Okay, so these are the two principal things that are occurring once you produce the exudate. In addition, the endothelial cells are going to do more than just endothelial cell contraction. They're also going to start recruiting actual immune cells from the blood. Principally, initially, they're going to start recruiting polymorphonucleocytes, okay, which are also called neutrophils. So I'll show one of these here. So here is a neutrophil in the blood here. Okay, and they have this very characteristic nucleus which has multiple lobes. So this is a neutrophil. I'll draw it bigger now. Okay, so here it is. And then its nucleus is separated into this bizarre shape like so, where you have multiple lobes, these 3 lobes generally, but it can be 4 or 5 lobes. Okay, and for this reason, the fact that all different neutrophils end up with different number of lobes in their nuclei, they're called polymorphonuclear cells. Okay, and that's not going to fit in. Polymorphonuclear cells, or for short, just PMNs, polymorphonucleocytes. Okay, so poly means many, morpho means shape, nuclear means nucleus, so many-shaped nucleus. Okay, so I want to stress this is not multiple nuclei, it's one nucleus just in a bizarre shape. Okay, now these cells are phagocytes, and you can recruit them out of the blood into the extracellular fluid of this tissue, but they can't just pass through these spaces between the endothelial cells without help from the endothelial cells. So the endothelial cells have to put on their surface all sorts of molecules which can bind to molecules on the surface of the neutrophil and help the neutrophil extravasate, as it's called, and leave the vascular system and get into the tissue. Okay, and then these polymorphonuclear cells, they come in and start phagocytosing the pathogen, so eating it effectively and digesting it. Okay, so that's the acute inflammatory response there. Now, this is important in what's known as ischemia-reperfusion injury, which is what we're about to talk about. But before I just talk about that, let's talk about what CB2 receptors can do to this inflammatory response. So basically, you find CB2 receptors on the sentinel cells. So I'll draw one here, and I'm actually drawing it as the G protein coupled receptor there for some reason. Okay, so you find CB2 receptors on the surface of these sentinel cells in a tissue. You also find CB2 receptors on the endothelial cells, like so. Okay, so this is a CB2 receptor as well. And basically, if you activate these CB2 receptors, the effect is anti-inflammatory. Now, what do I mean by that? I mean that you stop these cells, these sentinel cells, releasing the pro-inflammatory cytokines. Okay, so activation of the CB2 receptor is going to inhibit the release of pro-inflammatory cytokines by these sentinel cells. In addition, activation of the CB2 receptors on the endothelial cells is going to mean that even if the pro-inflammatory cytokines have been produced, the endothelial cells are not going to respond to them. So it blocks endothelial contraction, which blocks the production of the exudate, and it blocks this extravasation, as it's called, of leukocytes. Okay, principally initially polymorphonuclear cells. Later on, you'll start recruiting more fancy leukocytes like macrophages and eventually T and B cells potentially. Okay, right. So basically, CB1 receptors block the inflammatory response. They have a very powerful anti-inflammatory effect. Now, how does this tie in to the role of endocannabinoids that we've been building up, which is in ischemia? Well, potentially it has a role in protecting you against ischemia reperfusion injury. So let me just explain what ischemia reperfusion injury is. So basically, when a portion of tissue undergoes an ischemic period, okay, generally the damage to the cells of that tissue doesn't actually occur in the ischemic period. Generally, it actually occurs in the reperfusion period. So after the blood flow comes back, then the cells start dying in that reperfusion period. Okay, so generally it isn't the ischemic period in which the cells actually die. It's the reperfusion period that the cells actually die. So why is this? What happens in the reperfusion period? Well, basically, following an ischemic period, so let's draw a little flow diagram for this. So you have an ischemic period initially, which is a period where not enough blood is getting to an area, or potentially no blood at all is getting to an area. Okay, then what happens is you return blood flow back to normal in that area. What then happens basically is that the cells of that tissue start producing bad molecules when the blood returns, okay? Specifically, they start producing reactive oxygen species and reactive nitrogen species. So let me just give you some examples of reactive oxygen species and reactive nitrogen species. So we'll start off with reactive oxygen species, ROS for short. Okay, so the key example of a reactive oxygen species is superoxide. This is a very evil molecule, a very dangerous molecule. Okay, now this is written as O2 negative and then dot here. Okay, now let me discuss the actual structure of this with you. So basically it is an oxygen atom single bound to another oxygen atom. Now normal dioxygen, where you would normally have two oxygen atoms bound together, that's an oxygen atom double bound to another oxygen atom. In superoxide, the two oxygens are single bound to one another. Okay, so there are two electrons in here, one from this one from this one. So let's color code this. So this electron can come from this red oxygen here, and this electron can come from the blue oxygen over here. Okay, now, then if we look at the other electrons on each of these oxygens, one of them ends up with 3 lone pairs of electrons, like so, and then the other one has 2 lone pairs of electrons, like so, and then 1 free radical. Okay, so basically, if I contrast this to the normal dioxygen molecule, the normal dioxygen molecule would look like so. Okay, basically, a normal oxygen atom has 6 electrons in its outer shell. Okay, so 2 of them end up in lone pairs. 4 of them end up in lone pairs in a normal dioxygen molecule. Okay, for each of the oxygen atoms. And then what happens is each of the oxygens provides 2 electrons into this double bond. Okay, so here are the 2 electrons for this double bond here. Okay, and I haven't done the dot cross diagram, but never mind. The dots can be for this one. Okay, and 2 of them come from the other oxygen as well. So you have these 2 single bonds here making up this double bond. Okay, so this is the structure of a normal dioxygen molecule. Basically, to create superoxide from a normal dioxygen molecule, what you have done is put in another electron. Okay, and this has caused one of these bonds here to break. Okay, and 1 electron has gone back to each of the oxygens. So 1 electron went back to this one, and 1 electron went back to this one. Okay, and then the additional electron that you put in stays with one of them, and that's how this one's ended up with 1 too many electrons. You can see that it's got 7 electrons, it should only have 6, so that's why this one ends up with a negative charge. And then the other one, that does have the correct number of electrons, it has 6, but one of them is unpaired and therefore is a free radical, and that's what the dot here means. Okay, now free radicals are extremely dangerous chemical species because they are extremely reactive. Okay, so that's why this is called a reactive oxygen species, because it is extremely reactive. Okay, right, so that's a principal example of a reactive oxygen species. Other examples of reactive oxygen species include hydrogen peroxide, H2O2, which has this structure here. It's basically water but with an additional oxygen in it. Okay, like so. And an example of a reactive nitrogen species is what's known as peroxynitrite, which I'll just show you. So peroxynitrite, which consists of 3 oxygen atoms with 1 nitrogen atom. Okay, so you have a nitrogen atom here, double bound to an oxygen atom, and then 2 other oxygen atoms, like so. And this one has then acquired a further electron via an ionic means and therefore has a negative charge. Okay, so basically, following reperfusion after a ischemic period, the cells start producing all sorts of horrible chemicals. Okay, these horrible molecules here, reactive oxygen species and reactive nitrogen species. In addition to this, what you start getting is an inflammatory response occurring as well. So this response that we've just talked through here starts to happen. The sentinel cells in this portion of tissue that has just been reperfused start releasing pro-inflammatory cytokines and start triggering the inflammatory response. This brings in polymorphonuclear cells, and polymorphonuclear cells start spitting out reactive oxygen species and reactive nitrogen species as well. Okay, the reason is to try and kill pathogens. Okay, so you create these horrible molecules and tip them over the pathogens is the idea. So if this portion of tissue has got a pathogenic infection, the idea is we can poison the pathogen out by producing loads of these very reactive molecules. Okay, of course the reality is that this portion of tissue hasn't got a pathogen in it, it's just been reperfused after ischemia. And basically then what happens is this leads to these levels going up and up and up. So if I draw this in a flow diagram, the ischemic period also causes inflammation. Okay, and the inflammation then leads to further increases in the reactive oxygen species and the reactive nitrogen species. Okay, and these molecules are then going to cause the cell death. So this is what leads to cell death. Okay, the high levels of reactive oxygen species and reactive nitrogen species, which just basically react with all sorts of molecules, DNA, proteins, lipid molecules, and just basically make the chemical reactions of life not possible anymore. Okay, so this is how cells die following an ischemic period because of ischemia reperfusion injury. Okay, so the idea then is that endocannabinoids working through the CB2 receptor might have a way of protecting this, might stand as a way of protecting tissues against this. Okay, so we've discussed that in a time of ischemia, cells start producing endocannabinoids. So that means that what's going to happen is that the CB2 receptors on the sentinel cells and the endothelial cells are going to be activated, and this is going to have that anti-inflammatory effect. And the idea is that this can reduce this portion of this flow diagram, stopping the inflammation that occurs in ischemia-reperfusion injury, and then it means that the damage that is caused is going to be reduced because you're now not going to have as high levels of reactive oxygen species and reactive nitrogen species produced, because you're not getting that influx of polymorphonuclear cells into the tissue, which are key producers of these reactive oxygen species and reactive nitrogen species. In addition, I told you that the CB2 receptor was actually present on heart cells and on liver cells and on brain cells. And what it seems to be doing in those cells is that it seems to be activating the expression of certain proteins that protect you against the horrible effects of reactive oxygen species and reactive nitrogen species. So it again seems to be initiating protection. So potentially this could be helping in these tissues, protecting the cells from ischemia-reperfusion injury. Okay, so that then is how this all starts to come together. Cannabinoids seem to be very important in ischemia. Okay, tissues produce cannabinoids in ischemia, and this seems to firstly vasodilate the arterioles and small arteries that lead to that area and therefore help to restore blood flow, but it also seems to have this effect in trying to reduce ischemia-reperfusion injury by inactivating the inflammatory response to some degree, and also in protecting the actual cells from the damaging effects of reactive oxygen species and reactive nitrogen species. Okay, and that now concludes our discussion of the role of the endocannabinoids in the cardiovascular system.